welcome to Biochemistry 101. A little bit of warning is in place here because the biochemistry we're looking at here, obviously what you're getting is a very simplified look at something that if you take biochemistry in university, you'll probably spend at least a full year covering in far more detail. Probably, in a sense, you could look at this as what Disney says in the Jungle Book as being basically the bare necessities. You are introduced to a lot of biological molecules, and a lot of it is just recognizing four key macromolecules and recognizing that they're made up of certain monomers. So a lot of it is, if you can recognize the molecule, give their functions, you're there. So this would be the first segment. These are just your objectives from the course outline you have. Gives you all the references, and of course there's far more detail available to you and other resources made available to you. So to begin, the four macromolecules that we're actually looking at here, we're talking about, let's get a pen going here. And what color do we want today? Let's go for green. Yeah, four macromolecules. I guess what I should say first of all is when you talk about a word like macromolecule, it helps to know a few little Latin roots. Macro just means big. So these are big sucker molecules, and the four of them, as I mentioned, that you need to worry about in Bi 12. Carbohydrates. Yeah, you might have to use your imagination with my actual writing with this tablet, but carbohydrates would be the one, proteins would be the second macromolecule, and then the third, lipids, which you might want to just think of as fats, and then the fourth, not nearly as scary as the name suggests, nucleic acids, of which there's only two, so don't worry too much about it. Each of those macromolecules are made up of what are called polymers. So poly just means many. So these are combinations of monomers. They're made up of basically, in the case of carbohydrates, building blocks of glucose, or in the case of protein, building blocks of amino acids. So polymers just means long chains, and much of the rest of this section on biochemistry is just recognizing the monomers that make them up. So macromolecules, I think you should be comfortable with the fact that we're talking about big molecules. Forget that. Big molecules. Monomers just means one, mono. So the single units that make them up. Basically, that's what the graphic is showing. You have a long chain, a macromolecule, but that macromolecule has to be understood as being formed or synthesized because of the buildup of smaller monomers. So, two key concepts, and the two key concepts are actually opposites of each other. Number one being that these macromolecules, as I've mentioned, are actually built up or synthesized by putting together smaller monomers. But at the same time, you have to realize that these big molecules, to be much use in biological systems, they're going to have to be broken back down or hydrolyzed into those monomers once again. So you have a chapter, for example, on transport across biological membranes, like the cell membrane. Large sucker molecules, macromolecules, aren't going to cross biological membranes. They're simply too big. So again, you're going to have to look at how these macromolecules are broken back down into their building blocks. Same thing with a chapter, let's say, on digestion. That's really what digestion is. Digestion is hydrolysis, breaking down large molecules that you consume in your diet and putting them back into a form your body can actually use. So it's very important to know what the monomers actually are for each of the four macromolecules. So as we've mentioned, the four macromolecules are carbohydrates, proteins, lipids, and nucleic acids, but the monomers that make up carbohydrates are going to be, in the case of the carbohydrates, we're going to be dealing with glucose, which is, of course, just a simple sugar. And you'll get to know its formula, and you'll get to know what it looks like momentarily. The monomer that makes up proteins are called amino acids. Amino acids. And the monomer that makes up lipids are a combination of three, why don't I write three in there? Three fatty acids, that sounds kind of cute, combined with one molecule of glycerol. And finally, the macromolecule, the nucleic acids, DNA and RNA, are actually made up of polymers of nucleotides. Nucleotides. And again, as I said, it's not enough to simply know the names of these monomers. You're going to have to be able to recognize them and then figure out how they actually join together to make up the macromolecule. So this is one of your actual learning outcomes. Notice these are the same ones that are listed in your course outline. I'm going to read it to you just to emphasize the point that there's a verb that is very important in each of these learning outcomes. Distinguish among carbohydrates, lipids, proteins, and nucleic acids with respect to chemical structure. So obviously the word here, distinguish, is the verb that I'm referring to, because it really tells you what you're going to have to be able to do. I mean, I will know if you know what you need to know, if you can actually look at two-dimensional representations of some of these molecules, like the ones that I'm showing here, and say, those are obviously carbohydrates. And again, realize that if you've never taken organic chemistry, you're probably shocked by these diagrams, and undoubtedly there's a reason for that, in that you're just not used to looking at them. So again, it's more a matter of recognition, not so much technically understanding that carbon is tetravalent and all of these lines. There's always going to be 4 lines coming out of a carbon atom. So for instance, in looking at this group right here, notice that carbon always has these 4 bonds. All that means is it's a shared pair of electrons, a covalent bond between carbon and hydrogen. But again, you don't really need to understand the chemistry in terms of this objective and distinguishing among them, as long as you can recognize these 3, glucose, fructose, and sucrose, as carbohydrates. Glucose and fructose, of course, are monosaccharides, because they're only made up of one monomer, whereas sucrose is a disaccharide because it's got 2 glucose units joined together by this glycosidic linkage. The second one here, you're supposed to be able to recognize or distinguish it as a lipid, because here you have this C3 molecule is glycerol joined to these 3 long fatty acids, long chains that really determine which lipid it is. And then again, at the bottom here, what you have is part of a primary structure of a protein, because each of these joined together, these carbon to nitrogen bonds, are called peptide bonds. It's where the 2 amino acids come together. So you've got one amino acid and another amino acid joined together to make a protein. And then finally, the nucleic acid would be showing this nucleotide. And all nucleotides are simply made up of a sugar, which is the number 2 here, a phosphate, PO4, and then one of those bases that you learned about in earlier science, adenine, thymine, guanine, cytosine. So that's basically what they're after with that objective. And what I intend to do here is just go through that objective in a little more detail so that you can actually recognize each of them. So looking further at this objective, realize right off the bat that the word carbohydrate, the name itself implies the elements that are in a carbohydrate. Carbo, what do you think that tells you? That's telling you, of course, that it contains carbon. Hydrate tells you that carbohydrates all contain hydrogen, and the 8 basically signifies that they contain oxygen. So these are hydrocarbons made up of carbon, hydrogen, and oxygen. Then there's something called the empirical formula, not to be confused with the molecular formula. So the empirical formula is the simplest whole number ratio of the elements in that molecule. So in the case of carbohydrates, we would say that it's CN, which of course is a variable, H2NON. So in other words, 1 carbon to 2 hydrogens to 1 oxygen. Where you see this formula is in the actual monomer of carbohydrates that you really need to know the molecular formula of, and that of course would be glucose. And glucose, I'm going to give you the molecular formula, C6H12O6. You might want to repeat that to yourself 12 times before you go to bed tonight. But again, the point is, if you look at those ratio of atoms in that molecule, you're going to see it's 1 to 2 to 1. In other words, it complies with the empirical formula. Most of you probably have a diet high in carbohydrates. Not necessarily a good thing, but one of the reasons we tend to have a lot of carbohydrates in our diets is that they supply a whole lot of rapid, I guess that's the key word, rapid, wow, it's not the nicest R in the world, rapid energy, quick energy, and that energy, of course, is readily available for cell metabolism. At some point or other, you've come across the word cellular respiration. Cellular respiration is fueled by the energy, usually in glucose. So, yeah, and then sometimes you eat carbohydrates and you're not using that energy right away, so at the same time it can be stored away for later need. Rapid energy and storage of energy. I'm not going to write the word energy because you can barely read what I'm writing here anyways, but the storage form in animals would be a molecule called glycogen, which of course is going to confuse a few people, glycogen, glucose. But glycogen is just a storage molecule of glucose in animal cells. It's stored in the liver, and whenever you need to have glucose readily available again for quick energy, that glycogen can be broken back down into glucose. At the same time, plants can also store energy, which might explain why we actually eat plants. A lot of grains, for instance. The plant storage form of a carbohydrate is called starch, and it's stored in those wonderful little organelles unique to plant cells called chloroplasts. Similarly, if you're going to distinguish between, say, lipids, proteins, nucleic acids, and the carbohydrates, you're going to have to first of all be able to recognize what they contain. So in the case of lipids, not surprisingly, they too are made up of monomers containing carbon, hydrogen, and oxygen. And the lipids contain or include a whole lot of fats, what we would call triglycerides or animal fats, oils, and even something that we would call waxes. And all of these are classified based on the degree of what's called saturation. I'm going to put saturation here, even though at the moment you probably don't have a good feel for what saturation is. Yeah, that's one of the ways you distinguish between fats, oils, and waxes, and also the length of those fatty acid chains. So they're produced obviously by both plants. We use peanut oil, or we use canola oil. All of those are oils made from plants, and obviously they're made from animals as well. So I don't know how the last time you fried something up in lard, but you probably put butter on toast, and butter of course is an animal fat product. All lipids are made up of monomers of one molecule of glycerol bonded to 3 fatty acid chains. And of course that's what you get here. You got the glycerol molecule here, that's glycerol, and then each of these is a fatty acid chain, and they can be any number of lengths. The thing I was mentioning about saturation just refers to these carbon-to-carbon double bonds. If they're all single bonded, then we would say they're saturated. If there's any carbon-to-carbon double bonds, then we would say they're unsaturated. By unsaturated means you can add more hydrogens to them and actually break those double bonds. Distinguishing proteins, The only difference in terms of the actual atoms that make up proteins from the others, the carbohydrates and the lipids so far, obviously it's still going to contain carbon, still going to contain hydrogen and oxygen, but the one you want to notice is nitrogen. So if you see a nitrogen in one of these molecules, chances are you're looking at a protein, because one of the main components of all proteins is the monomer, an amino acid. And amino acids all contain this amine group, the NH2. And that's where, of course, you get this nitrogen. When you're talking about amino acids, we tend to have what are called 20 common amino acids. There's certainly more. And 9 essential amino acids. The 9 essential amino acids are amino acids that your body cannot produce on its own from other raw materials, so you actually have to obtain them in your diet. This is important in particular if you've decided to give up eating steak and you've decided to just live on rabbit food. You have to be very careful that in your diet you are getting those amino acids from other sources. So obviously in the case of protein from meat, those are complete proteins that contain the 9 essential amino acids. Without those, you're going to actually have to have combinations, say beans with some other source like rice to get the amino acids essential to make protein. It's going to go back a page here. Actually, these doesn't actually show it here, but you can't see the word joined by, but amino acids are actually joined by what are called peptide bonds. And again, we'll say more about these momentarily, but for now just realize that when you join the amino acids together, you're going to join them into a whole lot of different shapes and sizes to form very large protein molecules. Some of them are important, like hemoglobin protein that carries oxygen in your bloodstream on red blood cells, or a protein like insulin, which controls your blood sugar. All of those are simply combinations of amino acids joined in unique ways. Before moving on to that last macromolecule, the nucleic acids, and the distinguishing characteristics of it, I'm going to sneak in another one of your objectives. List the major functions of proteins. We've just been talking about proteins, and proteins are very unique molecules that are unique primarily based on the sequence of amino acids that make them up. And that sequence is called their primary structure. These are their straight chain structures. Those chains have a way of folding in on themselves to create helixes or assorted 3D molecules, globular enzymes, for instance. When we do the chapter on enzymes, this becomes incredibly important because these enzymes are essentially biological catalysts which will determine the rate of chemical reactions or even make the reactions go within your cell. For now, I'm simply going to bless you with the table in terms of what these functions are. The proteins themselves are usually classified based on their role within an organism. I've just talked about enzymes, catalysts. Some of those enzymes would be amylase, lipase, lactase, trypsin. You're going to, in a chapter on digestion, you'll find out that you have salivary amylase, an enzyme in your mouth which can convert a disaccharide like maltose actually convert the starch polysaccharide into the disaccharide maltose. If you chew up that cracker long enough in your mouth, you actually begin to sense that it's getting sweeter. That's because the disaccharide, the sugar, has that characteristic. Similarly, lipase, this little guy down here, is going to be an enzyme which breaks down fat. Lactase, another enzyme which breaks down milk sugar. Some of you have a genetic mutation that doesn't allow you to break down lactase, and so you have to supplement your diet or watch what you eat. Likewise, trypsin is an enzyme that's in your small intestine that breaks down protein. Similar function of protein is transport, and I've already mentioned hemoglobin. Hemoglobin transport oxygen and a small amount of carbon dioxide in your blood. Myoglobin does similar things in your muscle tissue, provides rapid oxygen to your muscle tissues for cellular respiration. Another function of protein would be simply part of your immune system, because antibodies are proteins. You need to fight off disease. That's what antibodies are part of. Contractile proteins are what a lot of us think of when we think of proteins. We think of muscle, and muscle is made up of filaments, myosin and actin, which again are proteins. There's a regulatory function to proteins, and again, here we're talking about hormones. Hormones are molecules that are released usually into the blood and then act at some target organ. So insulin is released from the pancreas into your bloodstream and basically speeds up the removal of that glucose from your bloodstream into various cells or storage places like the liver. Glucagon, again, is a hormone. Adrenaline, human growth hormone. Lots of hormones, and a lot of them, especially in the chapter on reproduction, you'll find out that females tend to have a few more of these hormones than males. And there's usually, a lot of resultant physiological and psychological responses to those hormones. The last function that you should be aware of is simply the structural function of proteins, that they make up some tissues like collagen and keratin. So the last objective, or the last macromolecule that you're to be able to distinguish, is the nucleic acids. Here it's good to know that for our purposes there's really only two of them. And you've heard of these before, because back in Science 10, or Science 9 I suppose it was, you actually got to know DNA. We will abbreviate it, and you can use the abbreviation, but do realize that it actually stands for deoxyribonucleic acid. And then the other nucleic acid would be ribonucleic acid, or RNA. So of the two, the most obvious thing you will notice is that DNA forms what is called a double helix. Double helix would be like the one shown over here, two strands in a helical pattern held together by bases in the middle. And way back again, I guess it was Science 9, you learned that these bases are adenine paired with thymine, guanine paired with cytosine. So it's always A with T and G with C. The difference, of course, would be RNA. RNA is not a double helix. RNA is simply single-stranded. But in both cases, the monomer of nucleic acids is, as I mentioned, I think earlier, something called a nucleotide. And because DNA and RNA are different nucleic acids, the nucleotides in DNA are slightly different than the nucleotides in RNA. So at the bottom of the screen, you have a phosphate joined to the sugar deoxyribose, and then one of four nitrogenous bases. So those nitrogenous bases, this one is thymine, adenine, cytosine, and guanine. One of the four makes up the DNA nucleotide. When you are considering an RNA nucleotide, on the other hand, you still have a phosphate, PO4, but now the sugar is not deoxyribose, but ribose. And the bases guanine, cytosine, and adenine are the same, but there's no thymine. I always like to think of the word no thymine. So you will have instead a replacement nitrogenous base, and don't be afraid of the word nitrogenous, it simply means contains nitrogen. Uracil. So uracil is found in RNA, but not found in DNA. So there are a whole bunch of different RNAs, and there's even a different DNA. You might have heard of mitochondrial DNA, which is usually abbreviated mtDNA. This is only found, as the name suggests, in the mitochondria, and it's only passed on in the mother's genes. When you look at lineage, it's basically called the Eve hypothesis. We can trace back this mitochondrial DNA back to some, I'm going to put the word Eve in quotation marks here, some original mother a long time ago who came out of Africa, and whether this is our biblical Eve or not, the point is that it does seem to be passed on just in the mother's DNA. Then there are RNAs. There's something called messenger RNA, which carries a copy of the code from the nucleus into the cytoplasm of a cell. And then there's something called rRNA. You have to sound a little bit like a seal to get that one out. rRNA stands for ribosomal RNA, a major component of that little organelle called the ribosomes. And then there's something called tRNA. tRNA stands for transfer RNA, and this is what brings the amino acids to the ribosome during the process of protein synthesis. So both of these nucleic acids obviously have major roles in genetics. In my way of thinking, I like to think of DNA essentially as a recipe book. A recipe book. I mean, if you don't like cooking, then the recipe book isn't necessarily going to hit it off with you. Think of it as City Hall, the plans for the city. Everything has some kind of a blueprint. DNA is the recipe for every single protein in your body. It is incredibly important. But before it can actually be used, you actually need a little helper within the cell, and this probably is just the helper in the kitchen that helps make protein synthesis happen. But DNA itself is the master book that has the actual code. So DNA is in code. That code has to be translated. That's the function of RNA.